So I have a question. We haven't done a question in a couple of hours. I know it seems like we just got started, but we are hours into this. We want a cure, and we want to know when you think we can achieve that goal, just given what you've heard so far. I see almost half very optimistic. Okay, thank you so much for joining in on these polls. That is just great information for us to capture from everyone. So before we go on to the next presenter, I'd like to make sure that y'all can see my screen, right?
Yes.
Yes. I'd like to acknowledge Amgen and Ultragenyx as the sponsors of our virtual conference. Both have been supportive of NUCDF's family conference in the past, and it's a trying time for everybody right now, and supporting the foundation with doing a meeting like this, we greatly appreciate their sponsorship. So I'm going to introduce Dr. Laura Konczal. Dr. Laura Konczal is a medical biochemical geneticist at the University Hospitals of Cleveland. Dr. Konczal received her MD at Wright State University College of Medicine in Dayton, Ohio. While in medical school, Dr. Konczal spent a month away with Dr. Carrie Orbitz and Longo at Primary Children's Hospital at Salt Lake City, Utah. This is where she met her first patient with urea cycle disorders and was inspired to pursue a career in medical genetics. Dr. Konczal was mentored by Dr. Douglas Kerr, Dr. Sean McCandless, Dr. Charles Hoppel, and Dr. Arthur Zen. It was through their mentorship that she was introduced to the Urea Cycle Disorders Consortium, taking a role as a co-investigator beginning in 2008 and becoming a UCD site PI in 2021. Dr. Konczal serves as the site PI for the Ultragenyx mediated gene therapy trials for late onset OTC. She became the site PI for Ultragenyx mediated gene therapy trial for late onset OTC phase 3 in 2022 and is a lead global site. More recently, Dr. Konczal was promoted to co-investigator for the urea cycle disorders longitudinal study with Dr. Susan Berry and Dr. Margot Braylon. Thank you, Dr. Konczal. This is Dr. Konczal's first time presenting at NUCDF. And I have to say that Dr. Konczal was mentored by Dr. Kerr, which was one of our favorites. And thank you for joining us today. There was actually a memorial service for Dr. Kerr today. So Dr. Konczal, thanks for splitting your time and being with us.
You're welcome. It was really kind of a interesting full circle moment to have the person that brought me into the UCDC and to introduce me to urea cycle disorders, to have his memorial, and we spoke a lot about that today, and his involvement. So, and I also saw many of my mentors that you just mentioned today who are also at that service. So I am fortunate enough to be able to talk with you about all of the exciting future possibilities for patients with urea cycle disorders. There is a lot to be hopeful for. And I really think, like the question asked, that we are going to see potentially a cure in the next several years. And I just think that's really amazing because we really haven't even known about the urea cycle for 100 years yet. So I found a really nice sunny beginning because I think that's what we're looking at in the future. And I thought this really fit with the title, that we really have these really exciting up and coming therapies on the horizon. And I really do think that we have a bright future ahead of us. Today's current management strategies for people with UCDs, they're not ideal, but it's protein restriction, essential amino acid supplementation, low nitrogen metabolic formulas like Cyclonex, giving L-arginine or L-citrulline to drive urea production depending on the disorder, ammonia scavenger medications that we just reviewed, liver transplantation in specific cases, patients that present as neonates, patients that have a lot of crises that aren't stable, they're difficult to control on medications, and sometimes families elect just the cost of and the expense of the medications. There's concerns about an out-of-pocket cap or a cap in their insurance over the lifetime. So some people realize that over time, maybe this isn't sustainable and opt for liver transplantation. Or as their kids are getting older, who's going to continue giving them their metabolic formulas and giving them their medications over time. Sometimes liver transplant makes it easier for care later on. So, and then we still use the same medications in times of metabolic crisis that we've already talked about. So the downside to all of this is the diet isn't that easy to follow, especially when kids get older and they have more independence. Teenagers tend to fall off the wagon. They're with their peers. They want to feel like they fit in, and it can be very difficult during the teenage years, young adult years, or even during early childhood when kids are later childhood when they're in school. They may not feel full. They may be hungry because they're really not getting very much protein. And then in certain states, metabolic formula may not be covered. So there can be expenses, or at least it has to go through insurance, and there may be copays. The medications themselves, as I mentioned, are very expensive. There can be side effects. Liver transplant is definitely an option, but there's always risks. It's a major surgery, long recovery. You have to find a donor. There can be complications. People can risk death with this surgery. There's long-term use of anti-rejection medications and immune suppression with it, so you're more likely to get an infection. And then there's also always the risk of rejection or failure of the graft and requiring a second transplant. And it is the expense. And then the acute treatment, when you're in crisis with Ammonul, there are side effects to Ammonul. It's expensive. Not every hospital has these medications available. So if you're traveling, you could be in trouble. They may not even have D10. And then people use dialysis, but there can be complications. And even if you have all of these options available to you, death or permanent neurologic damage can still occur as a hyperammonemic crisis is unpredictable. Or in the case of transplant, there can be complications or rejection. So that leads us to looking for better options and more permanent options. And I wanted to be inclusive of all of the urea cycle disorders. I think there's a lot of focus right now on the proximal urea cycle disorders, especially. But I just wanted to mention that the trial for the enzyme replacement therapy for arginase 1 deficiency, pexilarginase, and I hope I'm saying that right. And then I did want to touch on a recent clinical trial that didn't end up leading to any therapies that we have right now, but it just kind of illustrates that these clinical trials are important, but it's not a guarantee that it will result in a usable clinical option. So those were the hepatic liver cell infusions, hepatic stem cell treatments. There were trials in the US, a couple of trials here. One was Cytonet, one was at UPMC. There was another HepaStem, which was hepatic stem cells that they were infusing into the liver in Europe. There's no results for that, but they were all terminated before they were ready to be completed. So, and then I will talk in a little more detail about our current therapies that we have available, the mRNA therapies, gene therapy trials, and then the gene editing therapies that everyone's talking about right now. So just to touch on the treatment for arginase 1 deficiency, it is an enzyme replacement therapy that is injected either through IV or under the skin. It was a phase 3 clinical trial that there's some familiar names on this list. Dr. Enns, Dr. Diaz are all people who were active in the urea cycle consortium as site PIs, and Dr. Enns still is. But this study showed a really great reduction of plasma arginine. So for people with arginase deficiency, they can't break down arginine, they get lots of arginine. It can cause delays, but especially it can cause mobility issues. It can cause spasticity of the lower extremities. So they saw a more than 3/4 reduction of plasma arginine, which is amazing. And that meant in over 90% of patients with these conditions, their arginine levels were normal. So the data and the results that were reported look great. And they were able to look at patient mobility. They had the battery of tests, the gross motor function measure. and they found improvement. But despite these positive findings, and this is just a good point, the FDA wanted additional support and did not give them approval. So they wanted more information to prove that there was clear clinical benefit. However, so as far as I could find, as far as I could tell, this is still kind of in limbo. But in Europe, the manufacturer, Medica Pharma AB in Sweden, is manufacturing, and this medication is available as Loargis is the name of the pharmaceutical drug name for the same enzyme replacement therapy. So I guess that's a example of how you can have all this data, but not quite get through the FDA so easily. So then the other example that I wanted to just point out is when I finished my fellowship in 2010, I heard a lot about hepatic stem cell infusions. It was the hottest thing at the time. Everyone was really excited about it. And the company that was doing it initially was Cytonet. I kind of have these in reverse order. So they did this human I'm going to trip over my tongue here, heterologous liver cells for infusion in children with urea cycle disorder. So basically, they're taking liver cells from other people, and they're donor liver cells, and they are taking them and injecting them into the portal vein that goes into the liver to attempt to get the hepatocytes, enough of them into the liver to have working enzymes. So whatever urea cycle disorder usually was OTC or CPS1, the more severe disorders, try to get enough of those cells in to give them a little more time so that they could get a little bit bigger and have a better outcome or better chance at a better outcome when they go through liver transplant. So it was a bridge therapy to just give them more time to grow and stabilize so that they could be transplanted after 8 weeks, because some babies are just so severe that they may not make it that long otherwise and be forced to do a really early liver transplant. And unfortunately, that went for 5 years. And after enrolling 10 patients, it was terminated and there's no data. So it didn't necessarily work. And the same thing happened. There was a similar trial at UPMC, and they were doing something similar, but they were irradiating part of the liver to encourage those human hepatocytes to take root and maybe grow into that irradiated region, and trying to use that as an alternative to liver transplant. That also was terminated after enrolling 5 patients. It was terminated in 2022. It had been open since 2011. They are not going on with that either. So these are not options, but these are examples of clinical trials, seem like great ideas, didn't quite make it across the finish line. Similar thing in Europe, there was a HepaStem trial, very similar, but they used hepatic stem cells that can kind of grow and mold. Stem cells can become the cell that it can kind of mold itself to what's around it. So they injected into the liver. And this study took place from 2014 to 2017. They had 5 patients in several European countries, but they terminated it with no results. So it's all kind of the same story. It doesn't look like that one worked. So that brings us to where we are today in our current clinical trials. And there are 3 categories currently. And I think every one of them is exciting and every one of them has potential. The first that I'm going to talk about is mRNA therapy. And I wanted to just review this. I know that probably many of you are very familiar with DNA and transcriptions, translation, and how proteins are made. But for those of us who may not be, just so that it's clear how mRNA therapies work, I wanted to go over the basics. So if you look, I don't know if you can see my little arrow, but if we're looking at the DNA, which is in our nucleus, and that's where all of our genes live, and our OTC gene is there, our CPS1 genes. So there is a specific code, as you know, and what happens is if you have one of these conditions, there's a misspelling in your recipe that your body has for making that enzyme. So to treat people with urea cycle disorders, we are trying to give them a working version of that enzyme, that gene that does not work or does not translate correctly in that patient. So for mRNA translation, basically we have our genetic DNA code, and making mRNA, you just pair your letters. The G's are paired with C's, the A's are paired with U's in the mRNA. The T's are paired with A's, and then you get this mRNA code that is carried outside of the nucleus to the ribosomes, where each 3-letter group codes for a different amino acid. And those amino acids linked together become an amino acid chain that is modified, folded, and becomes your enzyme. So the initial therapies that looked very promising were actually in mice, which is usually how it works. We want to try these therapies in animal models before attempting it in people. But the OTC activity in OTC deficient mice actually showed really great response to the mRNA use. And mRNA is, it's not stable for very long. So when you inject mRNA, it only lasts for so long before you have to do another dose. So what they were doing in these patients is they would take that mRNA code, skip the part where they already had the mRNA created. The body doesn't have to translate that from DNA. It's very similar to the vaccines that we use for COVID, the mRNA vaccines. So, but the mRNA in this case is the mRNA code for making the OTC enzyme. So that code, the mRNA is injected, and in the mice it goes in IV. You can I think all of the trials are injecting them IV, but it is targeted to the liver. So once the mRNA code gets to the liver, it is coded and targeted for the liver cells. The mRNA is taken up, kind of enveloped by that liver cell, and the mRNA is then, the code is read. It goes to the ribosomes in that liver cell, and it starts using that code to make working OTC enzyme. And that, if you give enough of the mRNA, you can get enough enzyme. The goal is to restore your urea cycle function, at least in part, enough so that you're more stable and can break down ammonia. Unfortunately, you have to get injections every so often for that to continue to work. And that may not be a big deal. I mean, people with diabetes inject insulin. It just becomes part of your regimen. There's people with PKU that inject an enzyme replacement therapy daily called Palynziq. So it's not without precedent. So currently, this is Arcturus is the company that is making this mRNA therapy, and specifically for OTC deficiency. It's in the phase 2 trial. And currently they're evaluating the pharmacodynamics and safety. The pharmaceutical name of the mRNA product for OTC, the OTC mRNA is ARCT-810. And they started enrolling pretty recently, less than a year ago in November of 2024. The goal is completed by 2026. And this is all public knowledge that is accessible by anyone. So I'm not involved in this trial. So this is just information that I was able to research and find. They only need 9 patients. So this is to finish this phase 2 part of the trial. They don't need that many people. They need boys or girls, anyone over the age of 12 who has a history of high ammonia that they were symptomatic with, or elevated glutamine that caused symptoms. They have to be on a stable treatment regimen for 28 days prior to enrolling, just so that when they make changes, they know that that patient was already on a stable dose and that things aren't jumping around. And then patients, if they've been through a gene therapy trial or has stem cell transfer, like from the CytoNet study or the HepaStem study in Europe, they have to wait 2 years before they can actually take part in the study. Or if they haven't had a liver transplant, they're excluded. So, and I mentioned a lot of this information, but what will happen is all participants will get 5 doses of the ARCT-810, the mRNA product, and they usually give it through an IV. And there's 3 different dosing levels, so they're trying to find the best dose to give the best result without safety issues. And they've already preliminarily figured this out in mice, so they have a good idea where to start. They will measure ureagenesis. We use this a lot in a lot of the studies. There's a radio-labeled nitrogen, so a radio-labeled isotope. And the way that we can figure out how much urea somebody is making is that nitrogen gets through the urea cycle and is urinated out as part of that urea structure. And these urea studies, we know the dose that's being given, we can see how much of that nitrogen made it through the urea cycle. The more nitrogen, that labeled nitrogen that makes it through, the more activity you have of your urea cycle. So when we're doing mRNA therapy, gene therapy, and eventually gene editing, we want to see that compared to before you were treated, your urea production is a lot higher. And it's part of the study, they're checking ammonia, glutamine levels, looking at how the drug is acting in the body, how long it lasts, that's the pharmacokinetics. And this is a short-term study, it's less than a year, it's 85 days. Patients do have to travel to Common Cures in Maryland to be able to take part. So moving on, I'll talk about gene therapy. And I'm pretty involved with gene therapy as a PI. I had reached out to the lead of the study just to get permission to talk about it. And I reached out a couple of times, I didn't hear back. So when I'm going to talk about it, I will keep it to just information that's public knowledge. People are still blinded in the study, at least my patients are. So I really don't have any unblinded knowledge anyway, but I can give you a general idea of how it works and some general observations, but I don't have the Ultragenics graphs from the posters that I can share. But the adeno-associated virus is the vector that I mentioned is different than what was used when Jesse Gelsinger had his gene therapy treatment in 1999. So what the AAV virus is, it's part of the parvovirus family. It needs to be associated with another virus to replicate itself generally. So a lot of times it globs on to the adenovirus to get into the tissues. It doesn't usually cause anyone's symptoms. So a lot of people are exposed to AAV viruses and they never know it because it doesn't make you sick. It's out there, people get exposed. And it's estimated that depending on the serotype, about 30 to 50% of the population already have antibodies. If you have antibodies, you're ineligible for these studies because you already have antibodies that will just neutralize them. So that's part of the screening for any of the AAV-mediated gene therapy trials is to make sure you don't already have antibodies. Most people with the most common antibodies are with AAV2. We're generally using AAV8, which has the lowest number. If you look at this graph, AAV8 compared to AAV5 and AAV2 has the least number of people that are immune to it or have antibodies. And this was from a hemophilia study because patients with hemophilia have gone through gene therapy for years. They predated treatments for metabolic disorders. But the reason for using these AAV vectors is you can add a promoter, target it to specific tissues. So in our case, we're targeting it to liver. You can easily add a working OTC gene, eventually other genes, CPS1, there's many possibilities. Once you have the technology, you can always pop in any of these enzymes that work in the liver. So basically when we give these AAV modified vectors to the patients, it's just being given in saline. So you have your very large dose of adeno-associated virus that's been engineered to be targeted to the liver, so that when you give it through the IV, it goes to the liver and you don't get, you try to minimize the effects to other tissues. And it becomes active, it gets into the liver cells and is translated there. And the goal is for this adeno-associated vector to start to live in your liver cells and the hepatocytes and continually make working OTC enzyme over time. And it doesn't incorporate into your own DNA. It's separate. It stays with the virus. So I guess if you think about when people had before vaccinations for chickenpox, people get chickenpox, it would stay dormant in your liver. And then years later, you develop shingles because it was always there and it was still alive, but it wasn't causing you symptoms. So that's kind of the goal. And the purpose of the studies now, we're in some long-term follow-up studies, is how long does it last? And we're still learning. But for the OTC gene therapy trials that currently are now close to enrollment, so I was part of the phase 1/2 trials where we found the right dose. There was 3 different doses that were trialed. We had a cohort that was prophylactically treated with steroids because even though adeno-associated virus is safer and doesn't typically cause people to be sick, you give a very whopping dose of it, and that can cause inflammation, specifically to the liver. And we did find that if you give people, and you don't wait for the liver functions test to change, you give them steroids prophylactically, and this has been presented and is public knowledge for gene therapies in general, you can keep acute hepatitis from occurring. If you get an inflammation of your liver, it can neutralize it. Your immune system is revved up and it can neutralize that vector, that AAV vector that has your OTC gene in it, and then you don't have as good of an effect. So we want to get on it right away and prevent that. So as patients in phase 1/2 were given the gene therapy, it was not blinded. They knew they got it. They were on high dose methylprednisolone for a while, for a couple months, a tapering dose over time. And generally it was shown that it was safe. Nobody had any problems with reactions to the gene therapy. They didn't really there was no major problems with steroid-induced hyperammonemic crises. Everyone did okay. And everyone continued on to roll over into long-term follow-up studies. So we're continuing to learn over time. And everyone has been doing well. Some people went off of their medication, their Ravicti, butanol completely and stayed off of it, had a normal diet. Some people had a reduced dose, so they were responders. The complete responders were off of everything, could have a normal diet. And then we had patients who did not respond. There was just a few of them. But everyone, responders are not continued in the rollover. And this is given one time. So the gene therapy is given to someone who does not have antibodies to AAV8. And then, but once you do give it, your body makes antibodies, so you can't do it again. And so this was just the poster, and then we're working on publishing the data, and it was a multi-center global study. And you could just see that there were 4 cohorts. The 1.7 times 10 to the 13th genome colonies per kilogram was used to be the dose used in the phase 3 trials as it had the best response. But we also had prophylactic oral steroids given. And I mentioned we had complete responders. We had responders were anyone who cut their medications by at least half and had at least a 50% increase in their protein tolerance. We looked at ammonia levels, we looked at glutamine levels. And then phase 3, which is a double-blinded placebo-controlled trial, was a goal of 50 patients. And we didn't quite get there, but it's close to enrollment at this time. It was close. And this was for anyone 12 years and older with OTC deficiency who had at least one symptomatic hyperammonemic episode. Currently, the data is being analyzed. Nothing's unblinded yet, so I really have no data that I can share anyway. But in observation, I would say the patients that I've seen, it just seemed everyone has some degree of benefit, whether it's being completely off of medication and have a normalized diet, to being able to take less of the ammonia scavenger and maybe eat a little bit more protein, or just in general, being on the same dose and the same dietary intake, but just being stable and not being in the hospital all the time. So I feel like it did help everybody in some way at different degrees, but I don't have the data yet to confirm that observation. So that's gene therapy. And then related but different is gene editing. So this is what everyone's talking about right now. And I think this is a good kind of it's a little simplified, but it gives you a good idea of what's going on in gene editing. So basically, this is going to be targeted specifically and individualized to the patient's mutation that we want to fix. So what they do is they make an mRNA, but they call it a guide RNA, and it will target right to where that gene is in the patient's DNA. And it has a sequence to snip it out with a tool called Cas9. So Cas9 is it's like a pair of scissors that snips out that sequence that you want to get rid of. And then you pop in the targeted DNA sequence that is being made by our guide DNA. So you end up with a working piece of, so we'll say OTC. So we have a working OTC sequence that is popped in instead of that non-working sequence. And then the goal is it goes into your actual own DNA. So it will stay with your cells. And as your cells divide, it will be translated and go to the new cells. And the goal would be that it would last. And you have this working OTC enzyme, and it may not hit every single cell in your liver, but enough to make enough enzyme to make a difference. So currently there is a gene editing clinical trial that's enrolling, and I am not involved in this one. So all I can tell you is what I could find that was available to the public, but it's for babies less than 9 months of age. And this is through IECure. And they are using AAV vectors to carry the CRISPR-Cas9 gene editing tools into the liver cells. So it's given just like the gene therapy was given through an IV. It's targeted to the liver cells, but instead of making a viral genome that's making the working enzyme, it's going into your own DNA and fixing it. And the goal is to finish this by next year. It just started, it opened to enrollment in April of last year. There's 3 sites, Children's Hospital of Colorado, UCLA, and Mount Sinai. So that's exciting. And there have been patients that have enrolled. I don't know their outcomes, but this is leading up to what happened more recently, just last month, when there was a baby who had personalized gene editing through a different mechanism at CHOP at University of Pennsylvania. And it was in the New England Journal of Medicine. And I don't know if his family is on the call. I didn't want to use any pictures of the baby without permission. So this is just what's in the medical literature and just the headlines. But it's very exciting that this is a baby who had severe CPS1 deficiency, had symptoms within 48 hours, had a severely elevated ammonia, 1700. Clearly, it was like in many of our other patients or children who have severe CPS1 deficiency, and was treated, had the standard treatments, had a honeymoon period where he was stable, but then started to become unstable with recurrent episodes of high ammonia, was kind of hard to control. But while they were treating him, CHOP had a IRB, a permission from their institutional review board to do a rapid application to the FDA for personalized gene therapy for an individualized medication. And it seems to have been pretty successful from what I can tell. So the timeline is pretty impressive of how they made this diagnosis and got to this customized treatment within 8 months. So basically, they got the diagnosis soon after birth that it was CPS1 deficiency. They created a cell line. Some of the patient's cells were used to make a cell line. I think they also used the dad's cells to do some of the studies. But they tried to figure out which of his two mutations would be the best to create, that they would have the most success to do the CRISPR-Cas9 gene editing on. They selected the dad's truncating mutation. They made a mouse model. They used lentiviruses to modify the genes to create cell lines with his specific mutation so they could test it outside of the body before giving it to him. And they looked, after they treated those cell lines with the customized medication, to make sure that CRISPR-Cas9, which is what the medication contained, did not cause any problems with other genes. And they didn't find any, they found some off-target changes, but they weren't nowhere that was clinically significant. The FDA approved it in one day. They submitted this investigational new drug application, and they did toxicology studies in a primate, tested in a mouse model, and then they made the batch for the child. And by the time he was 7 months, got the first dose of this investigational customized new drug, and got a second dose at 8 months, and was cranking down his ammonia scavenger to half, was able to withstand a couple of viral illnesses without having a hyperammonemic crisis. And from what it sounds like, it's been very successful. And the name of the medication, I believe, is based on the name of the child, but they called it K-ABE as an abbreviation. And so I just have the companies in here who will be producing these customized treatments, but it's really exciting. And instead of using an adeno-associated virus, they're using a lipid nanoparticle to target the liver and insert this CRISPR-Cas9 technology into the cells. So you don't have that immune response because it's not an infection. So it's similar to the lipid nano technology that was used for Moderna and Pfizer's mRNA vaccines. So it's very, very, very cool. And as I mentioned, it sounds like the child's doing well, as far as I know, and remains stable and tolerated both of those doses of the medication without a problem. And the fact that it's not viral, they could give 2 doses without having to worry about if he had developed antibodies. And they used a different immune suppression. Rather than using steroids, they used tacrolimus, and so that just in case. And apparently that went well. So this is our last slide. So just looking at the risks and limitations of gene therapy and gene editing, comparing and contrasting. So the risks, and this is just general knowledge for any AAV-mediated gene therapy, whether it's for OTC deficiency, Sanfilippo, which is in development and close to being it's being applied for FDA approval, or any of the hemophilia gene therapies. The AAV can cause a hyperinflammatory response. And because of that, you can develop an acute hepatitis, which is why we have to give those steroids. And we can manage it as long as we give prophylactic steroids, we prevent that from happening. Also, microangiopathy is an issue, and that just means there's in the small vessels, you can develop little blood clots. That has not been seen in any of the urea cycle related gene therapy, the OTC gene therapy trials. It was in an organic acidemia gene therapy trial that used a different adeno-associated virus, but it can be significant. And there have been a small number of patients who have actually died after getting AAV-mediated gene therapy. Mainly it was people who had muscular dystrophies with AAV9. They already had pretty significant cardiac involvement, and inflammation from the gene therapy caused that to worsen. And they were cardiac related deaths. There was a liver failure early on. But I think as we're learning more and learning how to manage things, we get a better handle on it. There's also the steroid side effects that can be an issue. So people gain weight. It can be a little trying in teenagers. It can increase your blood sugar. So people who are diabetic, it can be an issue. It's known that high-dose steroids may cause decreased bone density, especially if you're on it for a while. Some people are more predisposed than others, and that can increase the risk for fracture. And then though I mentioned that at CHOP for the baby that they treated with their nanoparticle gene editing, they used different immune suppressants, sirolimus and tacrolimus, so they got around that issue with steroids. So that could be a potential for future gene therapy trials to try that instead. And then for gene therapy or gene editing, there's always the concern about off-target effects. So we don't know that much about gene editing. We're early in the process. So is it possible that you could affect genes that you didn't mean to with similar sequences? Yeah, it's possible. They're trying to minimize that by having very targeted sequences, but it could happen. The other concern is with the traditional AAV vector-mediated gene therapies. Could the genome of the virus be integrated into the human genome, cause problems? Could you disrupt a tumor suppressor gene with either gene editing or gene therapy? Yes, that could happen. And then that could increase. We don't know that it could happen, but we can't rule it out. So it is a possibility. And could that increase your risk of cancer? Maybe. So we don't know for sure that that happens. It's just a theoretic possibility, but it's something we just don't know. And then once these things get approved, the cost may be a factor. I know when SMA gene therapy came out, it was $5 million per dose. And I mean, as far as I guess the prices come down to 2.5, but people are able to get insurance companies. I think it just depends on, you know, if they can see the value. And as I mentioned, if you use AAV vectors, you can only use it once because of the antibodies. The lipid nanoparticles get around that. And then we also worry about hepatocyte turnover. And the younger a person is, the faster your liver cells turn over, regrow. And if you use a gene therapy over time, could you lose that viral vector genome that has your working OTC copy? Or could you lose cells that had gene editing? Because not every cell's going to get touched. So we don't know how long these things will last, or if they'll last forever. We hope they last forever. And then also for women, we don't know anything about what could happen if you get pregnant, if you are pregnant when you had any of these treatments. So there's very strict rules that if a woman is going to take part in these trials, that they need to be on 2 forms of birth, very reliable birth control, or have an IUD. And because of this, it can, especially if a woman's young, it could delay her childbearing years for a while. She may have to put things off depending on how long the trial lasts. It's not forever, but we just don't have enough information. And then my last slide, I just wanted to mention Dr. Kerr, because today's been all about Dr. Kerr here in Cleveland. And he was one of the pioneers that worked with Dr. Batshaw to publish that first paper in 1983 about the use of sodium benzoate and L-arginine, and his name was on it. He was one of the pioneers in this field, also in mitochondrial disorders and energy metabolism. But outside of medicine, he was an activist, a leader in the community, and just a wonderful person. So I wanted to give him a little credit here.